Hello friends, welcome back to another video tutorial from Arun's Biology. This is the fourth lecture of the genetics of bacteria lecture series. In this video tutorial we will discuss about the F plus factor, F minus, HFR strain, F prime and episome, as well as we will discuss about the formation of HFR or high frequency recombinant and F prime. We will also give a concept of sex duction or F duction in bacteria. So let's begin the topic. So first, the terminologies that we should understand in the genetics of bacteria are the F plus factor, F minus factor, episome, HFR and F prime. To differentiate that, we should have a knowledge about these terms. So F minus, we will start with this F minus. F minus means a bacterial cell that do not have any F factor. The F plus cell will have a autonomous F factor or fertility factor. That's why it is designated as the F plus cell. What is episome? After integration, the F factor with the capacity of integration into the bacterial chromosomes are called as the episome. What is the HFR? So HFR is the integrated form of the F factor. It is defined as a bacterial cell that carries an integrated F factor is called an HFR or high frequency recombinant. So here you can see this F factor is integrated in this bacterial chromosome. As a result, it is converted into this HFR form. What is F prime? The modified F factor formed by anomalous excision through reverse integration from HFR is called as the F prime. So if this F factor, which is already integrated in the HFR form, is come out of this bacterial chromosome through errorful excision through reverse integration, that will be called as the F prime, and that basically contains the F factor along with some of the bacterial chromosome within it. Now we will discuss about the formation of high frequency recombinant strain of E. coli. So this is the diagram of formation of HFR, and here the F factor is covalently inserted into the chromosome by a site specific recombination between the homologous DNA sequence in the F factor and the chromosome. So this is the F factor containing A, B, C, D genes, and this is the bacterial chromosome in E. coli. If they found some homologous sites, they undergo homologous recombination, and as a result this F factor will be integrated into the bacterial chromosome that forms the HFR cell. Now the integration of F factor is mediated by short DNA sequences that are present in multiple copies in both the F factor and the bacterial chromosome. If there is no sequence similarity between this F factor and the bacterial chromosome, there will be no homologous recombination and HFR will not be formed. And F factor can integrate at many different sites in the bacterial chromosome whenever it found any similarity in the sequence. A cell that carries an integrated F factor is called the HFR or high frequency recombinant. In its integrated state, the F factor mediates the transfer of chromosome from the HFR cell to a recipient cell during the conjugation process. Now if there is a conjugation between this HFR and F minus, this F factor will also be transferred to the recipient cell. That's why it is called the high frequency recombinant. The cells separate before the chromosome transfer is complete. Thus only rarely will an entire chromosome be transferred from the HFR cell to a recipient cell, because most of the time the conjugation process stops before the completion of transfer of the whole of this HFR. Now we will talk about the formation of F prime in bacteria, and this is caused by errorful or anomalous excision of the F factor from an HFR chromosome that produces the F prime factor that carries the E. coli genes. Okay, here you can see this is the F factor with the A, B, C, D genes, and this is the bacterial chromosome in the blue color. Now when there is a excision, there is formation of loop, and during this loop formation, due to some error, some of the bacterial genes, here the leucine and threonine, get within the loop of this F factor, and during the excision process, which is errorful or anomalous, the F factor that comes out contains the bacterial gene, here the leucine and threonine. So this F factor is called as the F prime, as it contains the bacterial gene leucine and threonine. We will write F prime threonine leucine, and this is the bacterial chromosome after the separation process. The F factor ranges in size from those carrying a single bacterial gene to those carrying up to half of the bacterial chromosome. Transfer of F prime factor to recipient or the F minus cell is called as sexduction. So this can come as a definition. What is sexduction or F-duction? That is the transfer of this F prime from a donor cell to a recipient cell is called as sexduction. It occurs by the same mechanism as F factor transfer in the F plus and F minus mating, but with much higher frequency. The F prime factor can be used to produce the partial diploid state or mero-diploid state, because suppose this contains the leucine and threonine gene, and in the recipient cell, if it also contains the threonine or leucine gene in the wild type form or mutant form, it will come in a mero-diploid or the partial diploid state. Mating between the F prime threonine leucine with the donor cell in the donor cell and the threonine minus and leucine minus, that is they are nutritionally mutant for these two genes, produces the threonine minus leucine minus F prime threonine plus leucine plus partial diploid state. This partial diploid state are unstable because the F factor may be lost, or they produce stable threonine plus leucine plus recombinants through homologous recombination. So after entering into the recipient cell, as there is sequence similarity in the threonine and leucine gene within the recipient, it can form homologous recombination, and these two genes, either in the wild type state or plus state, will be transferred to the recipient bacterial chromosome. So this is all about for this video tutorial. Thank you for watching.